Well, we continue our lecture on biochemical engineering. On this lecture, we talking about bacterial growth. So we have to take at first basic and the practical aspects. Life is essentially connected with growth. Phenomena of growth can be studied scientifically from two different points of view. First view is in the level of the cell or the organisms. Here, the finely tuned cellular, metabolic and regulatory process underlying the increase in length and volume, division and replication are of interest. So what we interest here is division of cells and duplication. View B on the population level, here the increase of number of individual or the population size and its mathematical describing and modeling are of interest. Strictly speaking, this is a study of populations, but nevertheless the term growth is commonly used. This can be studied and modeled without a knowledge of the cellular mechanism. A basic approach in microbiology is to study the growth from viewpoint B. This is experimentally usually simple because growth propagation of bacteria can be very fast in comparison to higher organisms and requires only small culture volume flasks. The formulas for theoretical treatment are not really difficult. Simple development of growth equations. The following approach to develop the growth equation is not the proper mathematical one, but it is illustrative and easy to follow without having to understand differential equation. Imagine a single cell of bacteria species in a medium with all the nutrients, oxygen, optimal temperature and pH. Assume that the cell of this particular species doubled or divided under optimal conditions every 3 hours, as we saw in figure 1. We thus write for the doubling time also generation time T subscript D will be equal to 3 hours. To describe the increase of number of cells over the time growth curve, we have to find an equation that expresses the cell number as a function of time. So we need to write an equation which included cell number is function of time, meaning that we have one variable is time, and this is independent variable, and the other variable is the cell number, which is dependent variable. So as we saw in this figure, let us imagine that this is our cell here, and with all nutrients and oxygen it will be grow and divided. So as we mentioned before, one cell gives 2, and this one also gives 2, and this one also gives 2. So from 1 it goes to 2, it goes to 4, then we goes to 8, then we goes to 16, then to 32, and so on. So that we have to call this is duplication time. Following table present how will cell number and time correlated. So we need to correlate cell number with time. Knowing that TD here equal 3 hours, but this is for one type of organism. This TD is variable according to type of microorganism. So TD will be variable according to type of bacteria or microorganism we use. It is not constant at 3 hours, but here we take it as 3 hours. So in this table we see that at time 0 here we have a one cell, and we want to correlate this to 2 power 0 cell, and I'll tell you why I use 2 power 0, and it's clear that this one, in order to include here the logarithmic mean, to find mathematical equation to represent this equation. So at time 0, after 3 hours, after 6 hours, after 9 hours, as I told you, this is first duplication 2 cells, after 6 hours 4 cells, after 9 hours 8 cells, and so on. And this is the time passed since the beginning of the experiment. For convenience, we here use multiple of the doubling time generally. This is 1 cell, 2 cell, 4 cells, 8 cells. This is the number of cells after each division event approach of a new generation, generally n. And this is we called n, and this is we called n equal 2 power n. The same number of cells as above, but written in a manner that reveals the mathematical principle behind the increase of n, generally n equal 2 power n. What we want to say here we have 1 cell. After 3 hours we have 2 cells, so this cells will be different from the first, and this 4 cells will be different from this, and also 8 cells will be different from 4 cells, and from 2 cells, and from cells. So we have to name first generation or known as, will give us daughter generation, such as in, if you can imagine a parent, they have child, this child will carry the specification both of father and mother. So properties of next generation will different from the first generation. However, the recognized equation n equal 2 power n does not yet include other variables time. We need to correlate time for the growth curve. But what really is n, can be regarded as the number of division events or the ordinal number of the daughter generation. So n here is no more than ordinal number of the daughter generation. What generation we have to increase? So this is first generation, this is second generation, this is third generation. So n will be 1, will be 2, will be 3, according to how much division we have in our cells. At the beginning there is no any division and there is no daughter generation. At n equals 0, after 3 hours there has been 1 division event, and the first generation will be daughter generation has appeared. At n equal 1 we have the first daughter of generation. Because the number of division events increase with time, they can express as a function of time. Just ask how to often the doubling time td is contained and fits into time t passed since the beginning. So n will be no more than time over duplication time td. As we said, this is variable according to the type of microorganism used. So n will be t over td. Here n capital denoted to the population will be 2 power t over td, as we will see here in equation 3. If a growth would be begun with 5 cells, we are talking about 1 cell. But if a growth began with 5 cells, imagine in the same flask with 5 individual dividing cell or 5 separate flasks, each with 1 cell, the equation would be n equal to 5 2 power t over td. And this 5 is the initial condition at time 0, at t equal 0, n equal n naught initially, and this is denoted to number 5 here. So n equal n naught multiplied by 2 power t over td. Note that cell number in culture will not increase stepwise in a stable function. N naught goes to 2 N naught, goes to 4 N naught, goes to 8 N naught. But steadily, this is because cells do not divide synchronously. What we mean here that we have this cell give us this cell. So this cell give two cells and this cell give two cells. These cells may be more faster give this than this and so on. And that's what we name as synchronously. Some will divide earlier, some will later. So it always divided, but some will faster, some will slower. Only with special experiment tricks it is possible to obtain synchronous stepwise propagation over a certain period of time. Some like writing equation 5 without a denominator in the exponent and replace the reciprocal doubling time 1 over Td by another constant, the doubling rate. So V will equal 1 over Td, and this is known as doubling rate. Since we have Td variable according to type of microorganism, we instead use doubling rate V, which is equal 1 over Td. So this equation 5 will be n equal n naught 2 power T over V or Vt. So if you look here, 1 over Td, put it V, so equation will be then conform to n equal n naught T 2 power Vt. Also, this is sufficient to describe growth. So we correlate n with time. But we need to eliminate 2 here, which is not sufficient for our mathematical equations. The expression of exponential function with a basis of 2 is not common in natural science. A much more common are exponential function with the basis of Euler number, when we know that E equal 2.7. A mathematically important non-periodic number, so-called E functions. Mathematically, an E function is the genuine, natural, or the exponential function. Because a given exponential function can be expressed with any basis, we can also express equation 5 as an E function without changing its curvature. By definition of the natural logarithm, the number 2 can be written as 2 equal e to ln 2. We know that e with ln will go, so 2 equal 2. This converts equation 5 to here, n equal n naught e to ln 2, all of this power to t over td. So we will get ln 2 to this power, so will become e to ln 2 over td multiplied by t, and this we know that ln 2 over td will known as mu as a constant. It's kind of specific or species specific. Unchangeable characteristic, every bacterial species has a particular genetically fixed doubling time under optimal growth condition. The doubling time of a given bacterial species growth optimally can thus be regarded as a fixed value, hence we can define the new mu. So ln 2 over td will equal 0.693 over td will equal to mu. The new constant mu is termed the specific growth rate as K in chemical reaction engineering, and is often its unit is day minus 1 or hour minus 1 or minute minus 1 is the reciprocal of time. So our last equation will turn to here, n equal n naught e power mu t. Equation 6 and equation 10 reveals the connection that mu equal ln 2 phi equal 0.693 phi, and we know for a particular microorganism V equal 1 over Td. In former times, V and equation 6, 7 and 12 were given too much consideration in the theory of bacterial growth. Today they represent only a side aspect, sometimes even leading to a confusion. And scent evaluation of a growth experiment is possible without using V. Equation 10 and 11 are definitely the really important ones. Form of cell counting to optical measurement. We know that we can't use a colony counter to account sample of some volume of culture. So for the measurement and the plot of a curve n versus t, we would have to determine the cell number in a culture at the beginning and then after different time intervals. Because we cannot count all cells and in culture volume V, we'll take a sample. So if we imagine what we have here, our culture, I cannot count all cells here, so I'll take sample from here and denote as n sample, and this n sample of known volume, so n sample over v sample will be equal to optical density of cell. So we have to account them in a representative sample, a liquid, with the volume v sample taken from the culture. For instance, in a microliter placed in a microscopic counter chamber, the revealed cell number n sample, which is, this is the count, is only a tiny proportion of the total cell number n, and just as v sample is tiny proportion of the total cell number. So it is almost immediately intuitive that the ratio n sample over v sample will denote to ratio of n over v in some manner. So this ratio, the cell number per volume unit cell mil or cell per liter minus 1, is known concentration of cells, or we can name it as optical density. So n sample over v sample will equal to cell density. Hence the cell number in equation 11 can be referred to the culture volume V. The equation then expresses the increase in cell density, so n over v equal n naught over v e to mu t. And this, when you go back to this equation, n equal n naught e to mu t, and instead of n naught we can use n over v, which is n naught over v here, just by dividing on v. This equation converted to this thing, so which is also n sample over v sample equal n naught sample over v sample e to mu t. This opens a possibility to avoid time consuming and boring cell counting. Instead of counting cells, we can use optical density by a photometer with or at same wavelength. So n sample over v sample is proportional to the optical density. We can determine a proportionality factor or conversion factor A and get an equation. This factor, once determined, allows calculating the cell density from any measured optical density. So n sample over v sample will be equation, and instead of proportional here we add a constant multiplied by optical density. However, more measurement of a growth curve, we don't even have to determine such a conversion factor, because it disappears if we insert optical density as correspondingly a optical density at initial condition. So optical density naught is optical density at time 0 and optical density at any time. So A multiplied optical density will be equals to A multiplied optical density naught multiplied E power mu t. So A goes from here and here. Turns to optical density will no more than optical density naught multiplied exponential mu t. Once we find this optical density at initial condition, we can compute optical density at any time from equation 18. So we can thus simply follow the optical density by optical measurement and plot it versus time to obtain a growth curve. From this we can calculate mu of the bacterium. For data evaluation we can display the exponential curve as a straight line if we plot the optical density on a logarithmic scale. So here linear scale and here is logarithmic scale. Optical density with time, here optical density with time, and this is optical density at initial. So at any point here we can use optical density naught multiplied, so we can O d will equal O d naught e to mu t, and we can, and this is O d naught at initial condition or time 0. So in a practical consideration, optical density is measured in a spectrophotometer, which we will see in figure 3. Underlying principle is that most of the light scattered by cell no longer reach the photoelectric cell. So when we use a source of light, I'll explain here. This is the basis of photometer. So we have source light, then we have here our cuvette. So this cuvette will be in some form like this, and we will fill this with our sample. So when lights come, here we have a blank solution, or we can use different solution as I'll explain later. So if blank solution here, we turn it to 0, then put our cuvette which contains cells. So when light comes to cells here will scatter the light, and only the past one will give us the number of optical density. Sometimes cells are of different colors, and the colors will affect the reading of our photometer. So instead of we put here distilled water, we will put a growth medium to make it to 0. Then we read the value here. So we consider optical density of bacterial culture is thus primary, not an absorbance. And you know what's the meaning by absorbance. As in the case of dissolved dye, cells of many bacteria are almost colorless, and real light absorbance is marginal. It is therefore not correct, even though unfortunately common, to designate the optical density of culture an absorption. The most appropriate term would indeed be turbidity. Only strongly pigment cells such as phototrophic purple and green bacteria cause significant absorption of light in addition to scattering. So we will have to expect that when light comes and our cells here, so some cells will absorb light and some will scatter light. So one should be aware that the photoelectric cells does not know what causes the decrease in light intensity. We have two causes decrease in light intensity. One of them is absorbance and the other is a scattering of light when passing through the cells. So it just notices less light which is displayed as a number, usually an extension value, but even if the display has the label absorption, you know that you are dealing with turbidity. So the practical use of the photometer requires consideration of 4 points: the intensity of light scattering and thus the optical density of a culture depend on the wavelength. Each process will work on a fixed wavelength. So when we work with 600 nanometer, we must all do all our experiment at this wavelength. So the intensity of the light scattering and thus the optical density of culture depending on the wavelength. Scattering of a short wavelength such as of blue light is stronger than that of long wavelength such as of red light. You can choose any wavelength for measurements, but once you have chosen, you must keep this wavelength constant for all following measurements. This wavelength, e.g. 600 nanometer, should be indicated as subseparate, e.g. when I mean optical density as 600 nanometer, we mean that this optical density was measured at 600 nanometer wavelength. The optical density measurement by a photometer depends on the geometry of the light beam, the position of the cuvette, and the photoelectric cell, i.e. on the photometer model and manufacturer. So if you have two photometer, when you use one, you have to complete your experiment with this one. You cannot move from this device to other device. So you cannot change the model of the photometer within an experiment. One should not even change between two photometer of the same model. If you have two devices of the same model, also you have to work with one device only in order to decrease the difference between these readings. So number C, because you want to measure only the optical density caused by the cells, any light absorption caused by the medium, brownish yeast extract, whatever nutrients, has to be subtracted. So this is most simply done if you initially fill the cuvette with a sterile clear growth medium, place it in the photometer and adjust the displaced extinction value to 0. So here you subtract all absorption of light by the yeast extract or nutrients existing in the sample. Every subsequently measured value above this is caused by the cells. Verify from time to time again with a sterile medium that the 0 point has not drifted. Readjust if necessary. Also make sure your cuvette and place it always in the same orientation. Do not turn it the next time by 180 degrees. So after each reading you have to empty and rinse and dry as possible the cuvette for the next reading or for next measurement. Do not touch the clear window side of the cuvette. Fingerprints must be removed. Number D, the proportional between optical density and cell density exists only for optical density less than 0.4 approximately, because higher optical density values are no longer proportional to the cell density. So you have to dilute your sample if you expect that its optical density is no more than less 0.4. So you have to dilute your sample by a factor D and divide it on this factor when you try to get your final reading. So here this is optical density with cell density. You see that the optical density approximately is minimum at 0.4. Lowering that 0.4 you have, or greater than 0.4, you have to dilute ensure to get to this region. So you have to dilute with distilled water by a factor D, and after your reading you have to divide your reading over this D. So if you have measurement optical density 0.27, sample diluted by D equal 1 over 2, that means 1 mol sample mixed with 1 mol sterile medium, so optical density will 0.135. Now division by D, optical density computed is 0.135 divided of D, which is 1.4, it will be 2.27. As above, dilution would not have been necessary. Here is the same 0.27, so we don't have need to dilute. Here for optical density 0.54, sample diluted by 1 of 2, 1 mol sample, 1 mol sterile, so optical density will be 0.288. This will be divided on half or multiplied by 2, so it will turn to 0.576, and that means we have to dilute this sample. So in reality, the semi-log plot of optical density calculated versus T does yield just the theoretically expected straight line, but more complicated growth curve. The reason of this is that cell inoculated into new medium do not immediately start growing. What we mean here, optical density calculated versus T just yield just the theoretically expected straight line, but a more complicated growth rate. When we talk about growth rate, we know that we have log phase here, then growth phase, then stationary, then dead phase. So we need more time here, and this is no growth area, and the reason of this is that cell inoculated into new medium do not immediately start growing, which should pass a lag phase, then start to grow with their optimal mu and the value you are interested in. They must first restart or wake up or adaptive with a new environment, then start to grow. Their machinery for the synthesis of adenosine triphosphate or DNA or RNA or proteins, which had been turned down during their previous storage in the refrigerator, for instance. We know that use an inoculum. This inoculum was stored in the refrigerator prior to start the process. When put it in new environmental, it will have some lag phase to adaptive and to start to produce ATP or DNA or RNA prior to division of cells. And you may calculate the time needed until the fermented cell with an assumed occupational volume of 1 micrometer cube would completely fill swimming pool. Actually, we can distinguish between different phases of a culture and see in this figure for calculation of mu. Only exponential phases are of interest. So this phase is our interest, lag phase is not interest, and also death phase is not of interest. Stationary phase, we have a balance between cell and nutrients, so it will be constant, which means there is no change in optical density and there is no growth. So what we have here, just exponential phase, what we concerned with. So exponential phase is used to determine mu from two data points, optical density 1 and optical density 2, that you choose exactly on the fitted line. Do not use just two measured values, which mathematically are never exactly on the best fitted line. If you would, so you would not make use of the best fitted line. There are two possibilities to do that. First, an easy graphic way is to choose the two optical density values, such as optical density 2, optical density 1. Then the time distance directly equates to d or td. And we examine here. This is point and this is point. We put here, and this time will be td. And this is the first case. The second case, the mathematical approach is to choose any optical density T2 and optical density 1 and the corresponding T2 and T1 values again from the line. The equation 36a is used to calculate mu from this TD can be calculated. So from figure we can calculate DD, or from this figure also you can use T2 and T1, and from equation 36a, as we'll see now, used to calculate the duplication time. So mathematical development for the growth equation. As we know, initial condition we have n naught. After some time we have n. So n over t approach to n. And because the newly produced cell always add to the population, this new cell will add it to the initial cell. So I expect that change of dn over dt will be equal to mu t. And by separation of variables and do mathematical integration, we will get that knowing that separation of variables yielding 1 over n dn equal to mu dt, just integration, this will give us ln n and this will give mu t. So ln n equal to mu t plus constant. At initial condition at time 0 the n is no more than n naught, so ln n naught equal to mu t plus constant, so constant equal to ln n naught, so equation becomes to ln n equal mu t plus ln n naught, so ln n minus ln n naught equal to mu t. This is no more than ln n over n naught equal to mu t. Take exponential for both sides, so n over n naught equal e to mu t, and thus will give us n equal n naught e to mu t, and this is equation 3. So if we have at any time we can compute the number of cells or population of cells. There are two further important connections. From equation 28 we get the same expressions for mu as in equation 10, but a more intelligible manner. If the time that has passed is exactly the doubling time, we can write t equal td within this time span. So n naught has increased to 2 n naught, such that equation 28 yields ln 2 n naught over n naught equal mu td, and hence mu is equal to ln 2 over td as we explained before. Equation 27 allows connection or calculation of mu from any 2 points that we place on the plotted curve. According to the relationships 15, equation 27 can be also expressed as ln optical density minus ln optical density initial equal to mu t. For two optical density values here, we can put ln optical density 1 minus initial equal to mu t1 over ln optical density 2 minus ln optical density initial equal to mu multiplied t2. Thus the difference 34 minus 3 33 equations yield ln optical density 1 2 minus ln optical density 1. Ensure that ln optical density initial will eliminate here, so will be equal to mu t2 minus t1. We know that mu equal ln optical density 2 minus ln optical density 1 over t2 minus t1. As we mentioned before, from the figure we can put t2 minus t1 here and compute mu. So mu equal 2.303 logarithm optical density 2 minus logarithm optical density 1 over t2 minus t1. Note that the application of this formula absolutely need the log of the two optical density data points. If optical density has been directly plotted on a logarithmic scale, the values are not yet the logarithmic, only their graphic display is logarithmic, so you still have to form the logarithmic mean. And this is the end of our lecture today. I hope you to read it, and if you have any question, just send me your question.